Joining me for Control of Ventilation, based on Desjardins Cardiopulmonary Anatomy and Physiology, Chapter 9. So to begin, in order to fully understand control of respirations, you need to have knowledge of these three things: the medullary respiratory center, the pontine respiratory center, and that includes the influence that the pontine has on the medullary respiratory centers and factors that influence rate and depth of breathing. So we're going to talk about these things today. Let's of respirations. We're going to start in the medullary respiratory centers. Although knowledge concerning this subject is incomplete, it is believed that two groups of interconnected respiratory neurons located in the medulla oblongata are responsible for coordinating the rhythmicity of respirations. These are the dorsal respiratory groups and the ventral respiratory groups. Collectively, these respiratory neurons are referred to as the medullary respiratory centers. In the respiratory components of the lower brainstem. The lower brainstem includes both the pons and the medulla oblongata pictured here. So according to this picture, what's located in the pons? The pontine respiratory center and the apneustic center are both located in the pons, or upper portion of the brainstem. What's located in the medulla oblongata, or the lower portion of the brainstem? These are the dorsal respiratory groups and the ventral respiratory groups, as well as the central chemoreceptors. Indicated here, you can see that the DRG are very close to the midline of the medulla oblongata, while the VRG are to the outside, more peripheral, and the chemoreceptors are on either side of the VRG. Let's start by defining a few terms: dorsal and ventral. Dorsal means closer to the midline. Ventral means outside of the midline. So, which portion of the brain gathers information from the peripheral stretch receptors and relays it to the VRG? It's the DRG. It's now known that the DRGs, both dorsal respiratory groups, assimilate various factors that influence breathing, such as peripheral stretch receptors in the lungs, central chemoreceptors, and peripheral chemoreceptors. The dorsal respiratory group is a relay station and coordination station. So they coordinate all of the information and then pass it off to the ventral respiratory group. The ventral respiratory group determines rate, depth, and rhythm of breathing. As you can see, the ventral respiratory groups are located bilaterally on two different areas of the medulla oblongata. The dorsal respiratory groups are closer to the midline. The ventral respiratory groups and the central chemoreceptors are more to the outside. Figure 1.9 in your book talks about this. The ventral respiratory group, or VRG, contains specialized neurons. These neurons fire during inspiration and another set fire during expiration. When the inspiratory neurons are stimulated, a burst of electrical impulses travel along the phrenic nerve to the intercostal nerves, causing the diaphragm and airflow to expand and airflow to go into the lungs. When the ventral respiratory group's expiratory neurons fire, the inspiratory muscles relax. The lungs recoil and expiration occurs passively. This inspiratory and expiratory cycle repeats continuously and produces a rate of about 12 to 15 breaths per minute. So, I want you to think about what we just went over. At what approximate rate do the VRG's expiratory neurons fire per minute in a healthy adult at rest? The VRG produces a respiratory rate of approximately 12 to 15 breaths per minute by firing off their neurons for expiration. Until very recently, it was thought that the dorsal respiratory group neurons were primarily responsible for activating inspiration. However, now we know that the VRG neurons are responsible for triggering inspiration, and they also coordinate the rate, depth, and rhythm of breathing. Breathing completely stops when certain VRG neurons are suppressed by something like an overdose of morphine or drinking too much alcohol. Pontine respiratory centers are located in the pons and consist of two specialized groups of neurons known as the pontine respiratory group. These, when I went to school, used to be called the pneumotaxic center and the apneustic center. However, we now just call them the pontine respiratory centers. The pontine respiratory centers are thought to modify and fine-tune breathing coordinated by the VRG. They also help to smooth the transition between inspiration and expiration. When the apneustic center, or APC, is disrupted, as is the case when there's a tumor near the apneustic center, prolonged or gasping type of breathing occurs. This is called apneustic breathing or breath holding like phenomena. Under normal conditions, the apneustic center receives several different inhibitory signals: lung inflation reflex, Herring-Brewer reflex, and that suppresses its function and permits expiration. Otherwise, you'd take a breath in forever. Multiple factors that influence the rate and depth of breathing. However, two of the most important ones are the central chemoreceptors and the peripheral chemoreceptors. Other important factors that influence rate and depth of breathing are the Harry-Brewer reflex, also called the inflation reflex. It's generated by stretch receptors located in the visceral pleura and in the walls of the bronchi and bronchioles. These become excited when the lung overinflates. Signals from these receptors travel through the afferent fibers of the vagus nerve to the respiratory components in the medulla, causing inspiration to stop. So the lungs themselves provide a feedback loop to terminate inspiration. Instead of a reflex to control ventilation, the Harry-Brewer reflex appears to be a protective mechanism that prevents pulmonary damage caused by excessive lung inflation. The significance of the Harry-Brewer reflex in an adult at normal tidal volumes is controversial. It appears to have more significance in control of ventilation in the newborn. The deflation reflex. The deflation reflex is when lungs are compressed or deflated with an increased rate of breathing that is the result. The precise mechanism responsible for this reflex and making the respiratory rate speed up isn't known. Some researchers think that an increased respiratory rate may be due to reduced stimulation of receptors during the Harry-Brewer reflex rather than the stimulation of specific deflation receptors. But others don't agree. They think the deflation reflex is not due to the absence of receptor stimulation of the Harry-Brewer reflex, but because the reflex is still seen when the temperature of bronchi and bronchioles is less than 8 degrees centigrade. The Harry-Brewer reflex is not active when the bronchi and the bronchioles go below this temperature, but the deflation reflex still works. The lungs are exposed to noxious gases or accumulated mucus. The irritant receptors are stimulated. The irritant receptors are subepithelial mechanical receptors that are located in the trachea, bronchi, and bronchioles. When the receptors are activated, a reflex vagal response causes the ventilatory rate to increase. Stimulation of the irritant receptors may also produce a reflex cough, sneeze, and bronchoconstriction. Juxtapulmonary capillary receptors. These are also called C fibers and are an extensive network of free nerve endings located in the small conducting airways, blood vessels, and interstitial tissues between the pulmonary capillary and the alveolar walls. The C fibers located near the alveolar capillaries are called juxtapulmonary capillary receptors, or J receptors for short. These receptors react to certain chemicals and mechanical stimulation. For example, they're stimulated by alveolar inflation, pulmonary capillary congestion and edema, humoral agents like serotonin, bradykinin, lung deflation, and emboli. When the J receptors are stimulated, a reflex response triggers rapid shallow breathing. Peripheral proprioceptor reflexes. The peripheral proprioceptors are located in the muscles, tendons, and joint, and pain receptors in the muscles and skin. When stimulated, the proprioceptors send neural impulses to the medulla. The medulla, in turn, sends out an increased number of inspiratory signals. This may explain in part why moving an individual's limbs during a drug overdose or producing prolonged pain to the skin, like a pinch, stimulates a person to breathe. Sudden pain causes a short period of apnea, whereas prolonged pain causes the breathing rate to increase. The proprioceptors in the joints and tendons are also believed to play an important role in initiating and maintaining an increased respiratory rate during exercise. The more joints and tendons are involved, the greater the respiratory rate. Hypothalamic controls. Strong emotions can activate sympathetic centers in the hypothalamus, which can alter respirations. For example, excitement causes the respiratory rate to increase. Also, increased body temperature causes the respiratory rate to increase, and decreased body temperature produces the opposite effect. For instance, a sudden cold stimulus, like plunging your face into very cold water, can cause the cessation of breathing, or at least a gasp. Cortical controls. Although the breathing pattern is normally controlled involuntarily by the medullary centers, one can also activate a conscious voluntary control over the rate and depth of breathing, or choose to hold a breath, or take an extra breath. Have you ever held your breath for as long as you could underwater? Those are cortical controls. Aortic and carotid sinus baroreceptor reflexes. These are in response to an elevated blood pressure. They decrease heart and ventilatory rate. They also can respond to a reduced blood pressure. In the case of a reduced blood pressure, they will increase the heart rate and ventilatory rate. Couple questions to get you thinking. What may occur when the lungs are exposed to noxious gases and the irritant reflex is activated? Think about it for a minute. If you said increase rate and also cough, sneeze, and bronchoconstriction, you're absolutely correct. My next question, which respiratory reflex or center is triggered by strong emotions? Think back. If you said hypothalamic controls, you're absolutely correct. This is why you have a high respiratory rate when you get excited. Which receptors initiate reflexes in heart rate and breathing in response to changes in blood pressure, either low or high blood pressure? If you said the aortic and carotid sinus baroreceptor reflexes, you're absolutely correct. Now let's talk a little bit more in depth about the central chemoreceptors. Central chemoreceptors are the most powerful stimulus known to influence the medullary respiratory centers. When there is an excess concentration of hydrogen ions, H+, in the cerebrospinal fluid, the central chemoreceptors, which are located bilaterally and ventrally in the substance of the medulla, are responsible for monitoring the hydrogen ion concentration. In fact, a portion of the central chemoreceptors is actually in direct contact with cerebrospinal fluid. It's believed that the central chemoreceptors transmit signals to the respiratory components of the medulla by the following mechanism. First, as the CO2 level increases in the arterial blood, during like if you were hypoventilating, you didn't have a respiratory rate, the CO2 molecules diffuse across the semi-permeable membrane called the blood-brain barrier, or BBB, which separates the blood from the cerebral spinal fluid. The blood-brain barrier is super permeable. It means that CO2 can cross easily, but is relatively impermeable to hydrogen ions and bicarb ions. So it lets in CO2, but tends to keep out hydrogen ion and bicarb. As the CO2 moves into the cerebral spinal fluid across the blood-brain barrier, it forms carbonic acid by the means of the following reaction. CO2 plus water goes to carbonic acid. Carbonic acid dissociates into hydrogen ion and bicarb. Because the cerebral spinal fluid lacks hemoglobin and carbonic anhydrase, it has a relatively low bicarbonate and protein level. The overall buffering system in the cerebral spinal fluid is super slow. Because of this inefficiency in buffering, the hydrogen ion generated from the preceding reaction rapidly increases and is significantly influential in reducing the pH of the cerebral spinal fluid. The liberated hydrogen ions cause the central chemoreceptors to transmit signals to the respiratory component in the medulla, and that in turn increases alveolar ventilation, makes us breathe faster. The increased ventilation reduces the PaCO2, and so the PCO2 of the cerebral spinal fluid is also diminished. As the PCO2 in the cerebral spinal fluid goes down, the hydrogen ion concentration of the cerebral spinal fluid also falls. This action decreases the stimulation of the central chemoreceptors. So, neural signals to the respiratory components in the medulla also diminish. This causes alveolar ventilation to decrease. Let's talk about the peripheral chemoreceptors. The peripheral chemoreceptors are special oxygen-sensitive cells that react to reductions of oxygen levels in arterial blood. They are located high in the neck at the bifurcation of the internal and external carotid arteries and on the aortic arch. You can see this in figure 9.4. They are close to but distinct from the baroreceptors. Remember, we talked about those? So they're separate. The peripheral chemoreceptors are also called the carotid and aortic bodies. So what do the peripheral chemoreceptors do? Well, they increase ventilation by the stimulation of a P oxygen level of less than 60 torr. And ventilation is suppressed in the case of a P oxygen level of 30 torr. But they have other responses as well. They also can cause peripheral vasoconstriction, like vasoconstriction of the hands and arms. You'll see this with blanching. Increased pulmonary vascular resistance, systemic arterial hypertension, tachycardia, and an increase in left ventricular performance. In a patient with a low PaO2 and chronically high PaCO2 level, like in end-stage emphysema, the peripheral chemoreceptors may be totally responsible for the control of ventilation. This is because chronically high CO2 concentration in the cerebrospinal fluid inactivates the hydrogen ion sensitivity of the central chemoreceptors. That is, bicarb moves into the cerebrospinal fluid via the active transport mechanism and combines with hydrogen ion, thus returning the pH to normal. A compensatory response to chronically high CO2 concentration. You learned about that with blood gases in Chapter 7. However, this is an enhancement of the sensitivity of the peripheral chemoreceptors at high CO2 levels. So, finally, it's important to understand that the peripheral chemoreceptors are specifically sensitive to the PO2 of the blood and relatively insensitive to the actual oxygen content, or CaO2, of the blood. The precise mechanism for this exclusivity is not really understood. This exclusive PaO2 sensitivity can be misleading. For example, there are certain conditions in which the PaO2 is normal and therefore the peripheral chemoreceptors are not stimulated, yet the oxygen content of the blood is dangerously low. Such conditions include chronic anemia, carbon monoxide poisoning, like burn victims, and methemoglobinemia. At what point are the peripheral chemoreceptors initially activated? When do they turn on? When the blood level of oxygen is 60 torr. Now, at what point are those same peripheral chemoreceptors suppressed? When the blood oxygen level falls to 30 torr or less. Great job on remembering. Get your brain going. What would happen to a chronic hypercapnia and hypoxemia patient if you gave them too much fraction of inspired oxygen? So you have a COPD patient coming into ER and you throw them on 100% oxygen. This patient has a history of having too much CO2 all the time. What would happen to that patient? Well, if you said their respirations may slow or even stop, you're correct. They are running on their peripheral chemoreceptors and they need to keep their PO2 around 60 millimeters of mercury in order for their body to kick in and say, breathe, breathe. If you give them too much, their body just goes, I have enough. So the peripheral chemoreceptors don't tell the medulla oblongata, the medullary respiratory centers, to take a breath. So, I live in Seattle, but sometimes I visit Denver, the Mile High City. Which chemoreceptors would be stimulated first by that higher altitude, and why? If If you said the peripheral chemoreceptors would be the first to be stimulated when the PaO2 falls to 60 millimeters or lower, you're correct. Those oxygen molecules are all scattered out. I don't have as much gravity pushing them together, and it's harder for me to keep my PaO2 up. Let's talk about some other things that stimulate the peripheral chemoreceptors. An accumulation of lactic acid can lead to increased alveolar ventilation. This is primarily through the stimulation of the peripheral chemoreceptors. What happens is there's a production of non-CO2 acids like lactic acid. The arterial hydrogen ion concentration goes up. It stimulates the peripheral chemoreceptors, and those in turn stimulate the medullary inspiratory neurons. That results in increased alveolar ventilation. This can be seen in figure 9.8. Something else that also stimulates peripheral chemoreceptors is hypoperfusion or stagnant hypoxemia, when the heart just doesn't move the blood around. Also, increased temperature and nicotine and CO2 can all stimulate peripheral chemoreceptors. Peripheral vasoconstriction, increased pulmonary vascular resistance, systemic arterial hypertension, tachycardia, and an increased left ventricular performance can all happen. The medullary respiratory centers coordinate both involuntary and voluntary rhythm of breathing. The medullary respiratory center of the medulla receives neural impulses from several different areas throughout the body, evaluates and prioritizes the signals, and elicits a neural impulse to the muscles of ventilation based on the metabolic need of the body. To fully understand this basic concept, the respiratory components of the medulla, including the VRG and DRG and PRG, and the factors that influence rate and depth of breathing, like the central chemoreceptors, peripheral chemoreceptors, Hering-Breuer reflex, deflation reflex, irritant reflex, juxtapulmonary capillary receptors, and peripheral prioreceptors, as well as hypothalamic controls, cortical controls, and the aortic and carotid sinus baroreceptors all must be understood. Also, miscellaneous factors may also influence breathing, which include the sudden and prolonged pain and sudden cold stimulation and stimulation of the pharynx and larynx. So here's a review question for you. What is the term for an involuntary diaphragmatic spasm followed by a sudden glottic closure? If you said a hiccup, you've been doing your reading. So here's another question. Which portion of the brain controls the rhythm of respiration? If you said the pontine respiratory group, you're correct. Where are the peripheral proprioceptors located? They're in the muscles, tendons, and joint. And remember that they respond to people running. That's why they think that when you run, you have an increased respiratory rate. We've come to the end of our time together. Thank you for watching this presentation about the neural control of respiration.